Good morning, everyone. It's a pleasure to be here, and thank you to Shiman and Yang Wang for the invitation. It's always great to be here in Beijing, in China, and I'm very happy today to show you with you some data that we collect in my lab that revolve around understanding a little bit more about a particular form of heart failure with preserved ejection fraction, or HFpEF, and among these a particular subtypes, so-called cardiometabolic HFpEF. So I most likely don't have to remind to this audience, mostly for the early career investigator in the back of the room, that the clinical syndrome of heart failure is majorly defined and divided in two major forms: heart failure with the reduced ejection fraction, or HFrEF, and heart failure with preserved ejection fraction, or HFpEF. And these two clinical syndromes are basically divided on the number of these parameters, which is called ejection fraction. That doesn't mean much, and most importantly, it doesn't tell you much about contractile function, overall function of the heart. However, it's now known that a large deal of epidemiological, clinical, as well as pathophysiological data distinguish these two types of heart failure as completely different species. What is more important is that if for heart failure with the reduced ejection fraction, we have a great knowledge of underlying pathogenic mechanisms, we have several effective therapies to combat this syndrome. Unfortunately, the same does not happen for heart failure with preserved ejection fraction, or HFpEF, which I will submit that we are still in the infancy in understanding of the underlying pathogenic mechanism, and so far we have a lack of effective therapies for only one FDA approved to treat this million of patients worldwide. And for the few that still think that HFpEF is just a more benign form of heart failure, I think it's important to recognize that HFpEF itself imposes a highly economic and social burden with a rate of mortality and morbidity that is comparable to HFrEF, and most importantly, it will represent, and it does represent, a major form of heart failure that we will face in the year to come. So perhaps one of the complexity to study heart failure with preserved ejection fraction is that HFpEF, and slightly more than HFrEF, is a highly heterogeneous syndrome. There are many phenotypes out there stemming from different comorbidities. However, the phenotype that is most prevalent and definitely associated with the worst clinical outcome is what we other call cardiometabolic HFpEF, or obese HFpEF, which I like to define as the cardiac manifestation of metabolic syndrome in which several comorbidities, in particular obesity as well as diabetes and other metabolic alteration, generate the clinical syndrome of heart failure characterized by a preserved or slightly normal ejection fraction. So this cardiometabolic HFpEF, or obese HFpEF, is highly prevalent and really true represent, due to the widespread obesity worldwide, the major form or subtypes of HFpEF in the years to come. But like I hint to already, so heart failure is a syndrome. And by definition involves several organ and apparatus, including the heart, but other tissue and other organs cannot be neglected when studying the pathophysiological mechanism of this syndrome. Of course, there are each one of them affected perhaps differently, and I would like to tell you a couple of stories, one related to the heart, I'm a cardiologist ultimately, and another one related to another metabolic organ, which we believe are particularly important. So when we look at the heart, of course, over the past few years, we have established a sort of translational platform with preclinical model of heart failure with preserved ejection fraction that can mimic the majority of the feature that we observe in the clinical syndrome of cardiometabolic HFpEF. And using this model as well as other preclinical models, we focus mostly on understanding cardiomyocyte behavior in terms of lipid accumulation, mitochondrial dysfunction, ER stress regulation. And the majority of these mechanisms that we identified in mice or rats had a clinical reality check using human endomyocardial biopsy from patients with HFpEF hearts. So one thing that, looking at the cardiometabolic HFpEF, and let's say one of the questions that keeps me awake at night is, since cardiometabolic HFpEF is a syndrome dependent or driven by obesity, However, it's not the same of obesity itself, where many people obese do not have HFpEF. What could be the obesity-independent mechanism of cardiometabolic HFpEF? So in other words, how we could disentangle the obesity component and what are the effects of obesity itself versus what are the perhaps peculiar and specific mechanisms of obese heart failure with preserved ejection fraction. So we approach this from the angle of metabolism, and in particular we're interested in understanding the metabolic alteration in this condition. Is of course, we know a great deal of what are the metabolic alterations that occurs in HFrEF hearts with the different substrate, energy substrate, how are differently utilized by the failing heart. However, the same does not happen for cardiometabolic HFpEF, for which the breadth and the entire collection of metabolic changes, how different energetic substrates are utilized in obese HFpEF heart, are still largely unknown. And thought to be true, there is one study from David Cass' lab in Baltimore. They look at the metabolomics of the human heart with HFpEF as well as HFrEF compared to controls. However, as I told you before, we are interested a little bit in understanding what could be the obesity, the metabolic changes that occurs in obese HFpEF that are independent of obesity. And from this data set, that again provide a great deal of information about the metabolic changes that occurs in obese HFpEF heart, this question cannot really be answered, mostly because the use of the control that were significantly lean compared to HFpEF, the use of RV biopsy, and the study of a phenotype of obese HFpEF, which is very likely to occur in the United States with an average BMI with 40 plus that we barely see in Europe as well as in Asia. So what we try to understand now is a little bit, again, how we can distinguish what is the obesity component versus the specific peculiar changes that occur in the failing obese heart. So we pair with a group of investigators in Germany that characterize very well from the hemodynamic standpoint and clinical standpoint patient with heart failure with preserved ejection fraction. And in this case, we are also lucky enough to collect left ventricular endomyocardial biopsy from this patient and to really address what is the clinical molecular metabolic landscape changes that occurs in obese cardiometabolic HFpEF from the left ventricle. So as you can see here, so this is the pipeline that derived in the lab to provide the first, let's say, atlas and metabolic landscape that occurs in LV obese HFpEF heart. This is we had to face some technical challenges. As you can see here, this is the little LV biopsy from HFpEF patient. For some reason, this patient not willing to give us a bigger piece of the heart. So, but using even from such a small sample, we were able to derive using mass spectrometry approach as metabolomics, lipidomics information as well as proteomics. And what I'm going to show you here is just a couple of changes that we believe are peculiar and specific of obese HFpEF compared to obesity relative to central carbometabolism. This is just to introduce you the population. Again, we collected LV endomyocardial biopsy from overweight obese HFpEF patient compared with as well as overweight obese non-failing control. And this population checks all the hallmark of HFpEF characteristic. Patients with HFpEF are slightly older, they have increased prevalence of atrial fibrillation, they have worsening functional class, increased biomarkers of heart failure such as NT-proBNP, but most importantly, there were no differences in terms of large metabolic changes such as BMI, body composition, and diabetes. So we started with a proteomic analysis. So even though, again, obesity is a big component of cardiometabolic HFpEF, using these two groups, non-failing obese control and HFpEF obese, we were able to distinguish differences between in the proteome between obese non-failing and obese HFpEF patient. And even if these changes are not massive and are not so extended as occurs when you compare with the lean control. The constellation of the changes in the proteome of obese HFpEF were able to distinguish at least 4 subtypes within the obese HFpEF group, mostly based on association with clinical characteristics such as age, NT-proBNP, or degree of fibrosis. And of course, when you do proteomics or any kind of omics, the first thing you do afterwards is looking at the gene ontology. And we were pleased to say that the majority of the pathways that were downregulated within the proteome of obese HFpEF patient were related to metabolic terms, broad metabolic terms such as nucleotide synthesis as well as ATP metabolic process. So this prompted us to use the very same samples to look at the metabolomics of these hearts. And just the 2 major pathways of central carbon metabolism, glycolysis, you can see here on the left, this is all the enzymatic reaction that from glucose will end up to pyruvate. And on the top of the slide, you see all the metabolites or the major metabolites of glycolytic intermediate. And on the bottom of the slide, you can see the proteomic level of the enzyme that catalyze this pathway. And when we look at the changes in glycolysis in this obese HFpEF hearts versus non-obese control, you can see that there is an accumulation of the proximal intermediate of glycolysis, such as glucose 6-phosphate, glyceraldehyde 3-phosphate, which was related to a reduction of the end product of glycolysis, such as pyruvate. And nicely correlated also these changes in intermediate proximal glycolytic metabolites with the changes of the enzyme that catalyze the first steps of this pathway. After glycolysis comes TCA cycle, and when we look at the intermediate and the protein that compose the TCA cycle, one of the major differences were the accumulation of succinate, one of the intermediate TCA cycle. As you can see here, there was also reflected in some generation of energy metabolite in the ratio between succinate, fumarate, or fumarate malate. But one of the holy grail, one of the hallmark of heart failure in general is that heart failure has been portrayed as an energy deficiency condition. And we were also able to look at this in the HFpEF obese heart, and perhaps less than what has been reported for HFrEF, but also in HFpEF heart, we were able to see a reduction in ATP-ADP ratio, suggesting that perhaps not to the same extent of HFrEF, also in obese HFpEF hearts, there is a reduction in energy presence. So I hope I convince you that at least for human HFpEF, the condition, the association between distinguishing obese HFpEF versus not failing obese hearts mostly lies on glycolysis with the accumulation of proximal intermediates and all the other downstream changes occurring in metabolism. So, but like I said, HFpEF is more about just the heart. There are several tissues, several organs that are affecting this condition. And another one that we got to be particularly interested in is the liver. Not just because the liver is one of the major metabolic organs in the body, but also because there is a disease of the liver that mimics or at least has some commonalities with heart failure with preserved ejection fraction or in particular obese HFpEF. So what we said a while back was to hypothesize that, perhaps a little bit bold in this condition, that cardiometabolic HFpEF is the so-called muscle D or fatty liver of the heart. Indeed, these two conditions, metabolically associated steatotic liver disease or muscle D, fatty liver, and cardiometabolic HFpEF share a number of future. From the epidemiological standpoint, they have similar risk factor and also mechanism of cellular dysfunction lying on lipotoxicity of inflammation can occur in both organs. Just to give you one data about epidemiology, patients with fatty liver have more risk of heart failure. But when you break down what kind of heart failure they encounter more, it's striking to see that the majority of patients with muscle D have increasing heart failure with preserved ejection fraction events rather than HFrEF. Again, suggesting that perhaps these two conditions work on the common pathogenic mechanism umbrella. And what is interesting to me is that in the liver, there is a very nice and clarified studiation of the process where from a healthy liver goes to the worst case scenario of liver disease, such as cirrhotic, with the intermediate studiation that goes through a lipid accumulation or muscle D towards a more fibrotic inflammatory phenotype, MASH, and up to cirrhosis, where it is almost irreversible. And this lies on several mechanisms, such as inflammation and lipotoxicity, similar to what occurs on obese or cardiometabolic HFpEF. What we miss in HFpEF is really this early stage of intermediate stage, where lipid accumulation and inflammation are perhaps reversible, and we can use this information for perhaps preventive strategies. So we are interested then to understand a little bit more about this conundrum between muscle D and cardiometabolic HFpEF under the hypothesis that these two organs talk to each other and there is a common liver-heart crosstalk that occurs between these two organs in the context of cardiometabolic disease, in the context of cardiometabolic disease. And what we need to approach here, what I present to you now, is a more system biology approach, try to understand and uncover what kind of candidates can mediate this liver-heart interorgan crosstalk in cardiometabolic HFpEF. So one of my students, Stefano, is a computational scientist, started very easy. We look at our preclinical model of HFpEF. As you can see here, looking at the liver histology, these mice have an element of fat accumulation. There is some disruption of cellular architecture. There is accumulation of neutral lipids in the liver. These mice do not have the inflammatory fibrotic phenotype, liver phenotype that we might see in the MASH or other more nastier, let's say, liver model, but definitely there is an element of muscle D or accumulation of lipids in this tissue. And what he did was to do RNA sequencing from the same mice of the heart and the liver, and as you can see here on the left, this is the constellation of the cardiac differential gene expression genes in the heart. There are a number of the heart that are down or upregulated in heart failure with preserved ejection fraction in the heart of these mice. And he did the same on the liver. So this is the differential gene expression in the liver of HFpEF mice, but in this case, what he did was to restrict the analysis to what we perhaps call improperly secretory differential gene expression, so secretory genes. So these are only the genes that, according to UniProt, encode for a protein that is secreted into circulation. So it's secreted in the bloodstream. As you can see here, there is a limited number of these, but there are some gene encoding for secreted proteins that are differentially upregulated or downregulated in the liver of HFpEF mice. And what Stefano did was to generate these maps of correlation between transcription of the heart and the liver. So he used this B-weight mid correlation in which every column represents a specific differential expressed gene in the heart of the HFpEF mice, and every row represents a specific liver secretory genes in the liver of HFpEF mice. And measuring the strength of this correlation, so the association between a gene dysregulated in the heart and a secretory gene dysregulated in the liver, he generated this map in which, as you can see here, there are some genes that correlate high with some liver secretory genes that highly correlated with the reduced gene expressed in the heart, some that correlate pretty high with genes upregulated in the heart. And he used this information and test the hypothesis if what this kind of liver secretory genes can be used to identify potential mediator of liver-heart crosstalk in different data sets. And he started using this high mouse diversity panel in which there is available cardiac and liver transcriptome from mice fed with high-fat diet only. Again, this is the map that I showed before from the HFpEF mice. He did the same exercise in this high mouse diversity panel. These are over 100 inbred strains that are fed with just high-fat diet, again, to identify what could be the liver-secreted genes or liver-secreted protein that are unique for HFpEF. As you can see here, there are some genes that are only present in HFpEF, but not in the high-fat diet only. There are some genes that are more represented in high-fat diet, but not in HFpEF, as you can see here. And using this, let's say, this approach, we were able to restrict our candidates to about 38 so-called mouse HFpEF specific genes that are only present in HFpEF but are not present in a high-fat diet. Again, to disentangle exclusively the effect of obesity from HFpEF. Then we take these 38 secreted genes and we look at the human plasma HFpEF proteome, and among these 38, 7 were actually found in the plasma of HFpEF patient. Among these 7, then we test the hypothesis under the assumption that liver transcriptome should correlate with the protein secretion in the plasma. So among these 8 that were actually found in HFpEF plasma proteome, only 2, serum amyloid A1 and serum amyloid A4, were actually show a strong correlation between liver transcriptome and plasma proteome in NAFLD patient. And as you can see here, ASA1 correlate with terms gene ontology in the heart that are associated with extracellular matrix remodeling as well as inflammatory condition. We went back to the mice and confirmed that ASA1 is actually liver produced. This is the heart, the white adipose tissue, and the liver of the mice, and only in the liver we detect ASA1 protein going up, as well as in the plasma of these mice. What is ASA1? ASA1 is an acute phase protein that has been associated with a number of metabolic disease. And just to give you a little bit of information, this is our HFpEF Berlin cohort in which we measure ASA1 in the plasma of this patient, and this correlates nicely, quite nicely, with the distribution of adiposity in human HFpEF population. So this work, I believe, had give us the first hints of what could be some of the liver-mediated interactors between the liver and the heart in the context of cardiometabolic disease, HFpEF, and muscle D. And ASA1, perhaps we have a number of data that I have time to show you that targeting ASA1 can be beneficial for the heart in the context of cardiometabolic disease. So I hope I convince you that studying HFpEF involves studying metabolic alteration that perhaps pass through the interaction between major organ, in particular the liver, as well as adipose tissue, and end up in regulating a number of metabolic changes in the cardiomyocyte and beyond in the heart, which can perhaps reverted by lifestyle intervention such as exercise training or dietary pattern. So with this, I'll stop here. This is all the people that work in my lab in Berlin, my source of funding. Thank you so much for listening, and happy to take any question in the discussion.